Hello, everybody. My name is Mirta Milic, and I am from the Institute for Medical Research and Occupational Health, Zagreb, Croatia. And today I will tell you something more about the use and the potential use of the buccal cells in human biomonitoring and early disease detection. So let's start. Before we start to speak about the buccal cells, I need to explain some details about the oral mucosa by itself. So oral mucosa, as you can see here, is the mucous membrane that is lining the inside of the mouth. It has actually been divided in two parts. The first part is the lower part, the connective tissue, and the other part that is closer to the surface is called oral epithelium. Those two layers or those two parts differ in two different things. The first is the resistance to abrasion, and the upper part is more resistant to abrasion. And you know that we are eating and chewing the hard food in our mouth, that we can even bite our mouth inside. So the cells need to recover, and this superficial layer that you can touch with your tongue always needs to be connected with the cells. So when you make a damage of your cell, the other cells need to replace those cells, either from this layer or coming from the layers below. In order to make those cells more resistant to abrasion, they are passing one process that is called keratinization or cornification. So how does this process happen? It happens from the connective tissue. There is a small layer of basal cells, and those basal cells are the only ones that can divide or make a new cells. From that layer, the cells are becoming differentiated. So that is the other thing, how those two parts are differing, because differentiated cells are losing the possibility to divide. They can only differentiate, meaning that their cytoplasm is becoming more polygonal, they are more connected, and they are passing from one layer, stratum basale, to the other, stratum spinosum, to the third, stratum granulosum, and to the fourth, stratum corneum, that is actually connected with your with the surface of your mouth inside. So while passing those layers, they are getting more and more keratin inside. So the surface cells are the ones that are the most keratinized. So they are the one who are resistant to abrasion. If you want to check something more about that, you can also see this article. So why buccal cells? They are really easy to collect, and they, when you are sampling the buccal cells, this sampling is part of the non-invasive techniques when you are comparing them to the blood sampling. So they can be easily performed also in children. You don't need any kind of specific equipment to sample those cells, so you can use either toothbrush, cytobrush, or spatula. The method for collecting or sampling is actually cheap, and buccal cells are the first level of exposure of the organism, and they are first level of organism defense or non-defense against all the agents that can come or enter into your organism or body through respiratory tract. So that means the buccal cells are the first contact in inhalation, in allergies, in ingestion, especially from the food and the water that contains xenobiotics. And for example, if you are drinking or using some food or water that is rich with arsenic, arsenic is stored in keratinocytes, and the cells that are rich with keratin are the buccal cells and your hair. There is no need for cell culture and division when you are using the buccal cells, but the name buccal cells refers to all cells type that you can find in your mouth. I will explain later what does that mean, because you have also basal cells, and you can make some stem cell culture from the basal cells. And those stem cells can be used for gingiva destruction, bone destruction of the teeth, and eye epithelial diseases. We will talk today only about the exfoliated ones, not the gingiva ones. These buccal cells, when you are assessing the DNA damage from them, they are really informative because I have told you after the passing the basal cell layer, there is no division of the cells. So the DNA damage that has been made in the basal cells is not lost to the division, or new damage cannot be made. You need to know that those cells need like 10 days or 2 weeks, sometimes even 4 weeks to come from the basal layer to the surface. So you can measure the DNA damage in them from the recent exposure and from the chronic exposure. So not from the immediate exposure. You can detect different types of DNA damage, and there is also correlation with the damage from the buccal cells with the DNA damage found in the circulating lymphocytes from more invasive techniques of sampling. Because buccal cells have demonstrated a higher DNA damage, or some types of DNA damage in some types of diseases, buccal cells are having a possibility to be used as early biomarker of the disease. Today, buccal cells are used to test anti-cancer agents, the impact of malnutrition, and defects in DNA repair. Usually, there are 2 genotoxic assays that are used to assess the DNA damage in the buccal cells. And the first one is the buccal micronucleus cytome assay. Usually, we say that this assay is measuring permanent DNA damage. And the buccal comet assay. Usually, we say that this assay measures the DNA damage that is like a primary, but actually it measures usually the DNA damage of the basal cells. I will explain to you later why. But for both assays, it is important what I have told you already, that you need at least 10 to 14 days that the cells differentiate or pass through all those layers to the superficial, where you will collect the cells either with a toothbrush, with a cytobrush, or with a spatula. Sometimes some people have a different time of differentiation, especially if they are not chewing the hard food, or they don't have too many damage on the surface or inside their mouth. So sometimes those cells even need 30 days to come to the surface. So that means that studies that you will perform on the buccal cells cannot start earlier than 10 days after the acute exposure. If you have a chronic exposure, then you can collect the samples anytime. It is also important to wash those epithelial cells once or several times, like 3 times mostly, or in general. And what are the solutions that you can use for washing your mouth are either saline or some specific buccal cells buffer. For example, those ingredients for one typical one are written here. Or you can use water, but it should be either tap water or the bottled, but non-sparkling water. That is important thing. Why washing step? With this step, you are eliminating the microbes that you still have in your mouth that can resemble to the DNA of the buccal cells, and also to remove the non-attached cells from your mouth. Here are some types of the DNA damage that you can see in the buccal cells and if you're interested more in exploring how those cells differ, you can read also this manuscript. Why did this buccal micronucleus assay become interesting? There are already protocols for the micronucleus assay in vivo and in vitro. In vivo means to perform micronucleus assay in animals, but you are not performing that assay in the buccal cells, but you are performing it either from the blood or usually from the reticulocytes of the animals. You don't need no cell culture, and you are making the smears on the microscopic slide and examining them after the staining, usually under fluorescent microscope. But you are using the animals, and in the standard examinations and protocols, we are preferring not to use the animals to test the chemicals or some kind of agents. There is also in vitro protocol for micronucleus assay, and if we are talking about the human biomonitoring, you are using invasive technique of sampling the whole blood. You need a cell culture because usually those are the human lymphocytes that you are using in the whole blood, and they need some time to divide. So to finish this protocol or this technique, you need 72-hour cell culture, and you are assessing the DNA damage in the cells after the adding of after the first division of your cells, and usually after the adding of one chemical that is called cytohalazine B that stops the cytokinesis. So it allows the karyokinesis to appear, so that from mother cell to new daughter nucleus are made that are sharing the same cytoplasm, and you are checking this their shared cytoplasm for the DNA damage. The guidelines for both in vitro and in vivo assay exists, and they are internationally accepted guidelines that have been made or validated by the OECD, that is Organization for Economic Cooperation and Development. This organization has made a really like a set of guidelines for chemical testing that you can also use for other agents that are not chemicals to test their toxicity and DNA damage assessment. So the protocol for the micronucleus assay in vitro has the test guide, OECD test guide number 487. And in this assay, as you can see also in this picture, micronucleus, because that is how this cell, this assay has got their name, is the part of the DNA that was not shared between two daughter cells in their nucleus, and it has this shape, round shape, and it is located in the shared cytoplasm outside of those two daughter nucleus. This micronuclei can originate from acentric chromosome fragments, that means from the loss of part of the chromosome, or breakage of the chromosome, or the loss of the whole chromosome. If you are using specific fluorescent probes, you can also see if those two micronuclei or one micronuclei is actually done or made by the loss of the entire chromosome. Then you have, if you are using centromere probes in, for example, in a fish, you will have two centromere signals. So that means that the entire chromosome is lost, and that is a part of the micronucleus. Or if you don't have the centromere signal, that means that you have only part of the chromosome that is in your micronucleus. So this assay can detect the clastogenic or anergenic activity of some kind of substance. What does that mean? Clastogenic means that the chemical or the agent has the influence on making chromosome damage. But if it has aneugenic influence, that means that its influence is on mitotic spindle. So you can use cytohalazine B chemical or not, I have already told you. But usually we are using cytohalazine B and assessing the DNA damage after the first division in shared cytoplasm. This assay has become now a cytome assay because besides the micronucleus, you can measure either different types of DNA damage, and that is nuclear, but that is a biomarker of amplification of the genes, and you can see how it looks like here. So it is similar to micronucleus, but connected with one nuclear, with one daughter nucleus. You can also see the nucleoplasmic bridges. That is actually a biomarker of dicentric chromosome that is shared between two daughter nucleus. And you can also measure the cells or the frequency of the cells in apoptosis, that is a programmed cell death, when the cell has too much damage that cannot be repaired. And instead of making a big pool of non-stable, genomically unstable cells in the future, the cells by itself goes to program death so that this genomic instability doesn't increase in your system. Or you can measure the cells that are in necrosis. There are articles, or there is enough evidence that have correlated the DNA damage between the lymphocytes, buccal micronucleus cytome assay, with the lymphocytes and the buccal micronucleus cytome assay. And here on this graph, you can see this linearity. Usually for healthy population, the frequency for on 1,000 binucleated cells is up to 13 or even 15. For the buccal micronucleus, that frequency is really lower, as you can see, let's say up to 2, depending on each laboratory. So this is like the meta-analysis or statistics on 5,000 individual data from different labs. It has been shown that either lymphocyte micronucleus frequency is higher, but when we are talking about cancer, non-cancer diseases and cancer, interesting is that the ratio of micronucleus frequency in people with cancer disease and cancer was much higher in exfoliated buccal cells than in lymphocytes. And the strongest association for the buccal cells was when micronucleus was measured in the cells from the same origin as the cancer. And that means that a good prediction for cancer risk was seen in different types of the oral cancer in the case of the buccal cells. There are evidences, even from 2011, that micronucleus frequency in peripheral blood lymphocytes is connected with the elevated level of the cancer risk. Those studies, this has not been confirmed in general for every type of cancer for the buccal cells, but still with the new evidence, with the new manuscripts, I think that this will be also confirmed. We are talking about all time about the biomarkers, but I am not sure, do you know what is the definition of the biomarkers? So let's say a few words about that. So biomarkers are used for human biomonitoring in general, also for general and for working populations. And they present a link between biomonitoring tools and understanding the mechanistic pathways that are connecting exposure to some kind of individual health risk or disease risk. Based on different strategies of the measurement, those biomarkers can be divided on biomarkers of exposure that reflect the exposure itself, on biomarkers of the effect that are connecting or measuring the consequences of that exposure, and susceptibility biomarkers that are measuring or assessing the susceptibility of each individual to predict the intensity of the effect. So after this explanation, can you maybe answer in what of those three categories of biomarkers are micronucleus and Comet assay, or either micronucleus assay? I will give you like 3 seconds. And the answer will be on the other slide. So the answer is that this micronucleus assay, and later I will tell you about the COMET assay, they are both biomarkers of the effect. So they are involving quantifiable changes in biochemical and physiological parameters. And if you want to learn more about different types of biomarkers and what types of the biomarkers can you use, you can check this page, and also our other talks Learn for You materials. So this is a scheme of the buccal micronucleositome assay protocol. So it is really simple. Participant need to consent to give their example, and then you need to collect their example. You need to, before the sampling, you need that that individuals need to wash their mouth. So then goes the cell separation and the removal of the bacteria's microbes and cleaning of the sample. So then after that, you are counting the buccal cell number, and you are preparing the microscopic slides and staining them, and you are making microscopic analysis. You can do it under the bright field, but usually you are doing it under the fluorescence, with the fluorescence microscope. What is the difference actually between the lymphocyte micronucleus and the buccal micronucleus? It has, again, the micronucleus. It can measure also the cells without the micronucleus, so that means without the DNA damage. There is a nuclear bud and a structure that we did not show here that is called broken egg. So those two structures, as I have already tell you, that in differentiated cells, you don't have the cell division anymore. So this DNA damage is not the result of unrepaired DNA damage after the first cell division. Why this first cell division is important in lymphocytes? Because in that cell division, all those 5 mechanisms of DNA repair are active, and they are repairing the DNA damage. And actually in that shared cytoplasm in lymphocyte micronucleus assay, you are measuring the DNA damage that is not repaired. Here, there is no cell division. So this is the damage that has been made in basal cells. So the origin of the broken egg and nuclear buds is a little bit different than in the lymphocytes. Binucleated cells are again not a result of the shared cytoplasm, but actually they are formed from the basal cells, and they are biomarker of some kind of cytokinesis defect. In this assay, you can also measure, as in micronucleus in lymphocytes, apoptotic and necrotic cells. But here, you can measure early apoptotic cell effect and late apoptotic cell effect. You can see how those structures look like in the nucleus of those cells in the buccal micronucleositome assay. And you can measure the early necrosis in the shape of pycnotic cells and late necrosis. This is a cell that doesn't have a nucleus anymore, but it looks like a ghost cell. So if you want to learn more about the protocol, you can check also this manuscript. So what is important for the buccal micronucleositome assay? There are some rules that are similar for measurement of DNA damage, like for the lymphocyte micronucleus assay, and that is that if you are counting micronuclei, the size or diameter of the micronucleus should be minimum or maximum 1/3 of the main nucleus when you're comparing it to the main nucleus until the 1/16th part. It should have the same staining and texture, and it should be in the same focal plane as the main nucleus. This technique started in 1982, and this was the first published article. that has examined the effects of the chewing metal quick. So that means some types of the smoking or tobacco use in lymphocytes, but also in cell cultures. And the problem from that time until now was also the staining, because people have used the DNA stains, but not DNA specific stains. And you can see on this picture how difficult it was, because also the microbes were stained because they also have the DNA, how difficult it was to conclude what is actually the micronucleus and what are the microbes and other artifacts that have been counted. Here you can see different staining. This is one fluorescent stain. So that also the question is, what is here a micronucleus? So it is really hard to answer on this question. Here you can again see what were the problems when you have a lot of bacteria, and to check which filter, which staining to use. And you can see what were the problems, if you are interested in that, in those two manuscripts. But today, there is a big group of scientists that is gathered in human XCell group. They are called like this. And they have validated and made a standardized protocol for the buccal micronucleus comet assay, and have concluded that Folgen fast or light green staining is actually the best ones to use and to get more precise results. So if you want to see the gallery of the pictures and see how the damage can be measured or seen, you can check also this manuscript. We will now, from that manuscript and from that staining, you are actually using chloric acid, then Schiff aldehyde, and then fast or light green staining, and you are under the fluorescent microscope can see all those structures. So if you have checked already that manuscript, or you, without checking, maybe you can answer what are the types of the cells and DNA damage that you can check with this type of preparing your slides. So I will give you like 5 seconds, and then I will show you the results. So on this cell slide, you can see, you can measure in this assay the basal cells that I have told you that have still the possibility to divide. They have a big nucleus, and ratio of the nucleus comparing to their cytoplasm is really, let's say that this cytoplasm is really small. Then in differentiated cells, you can still have larger nucleus that is actually shrinking a little bit, but the cytoplasm is becoming much, much larger and polygonal. This you can see the binucleated cells. Here is the early apoptosis, and this is the nucleus that has the condensed chromatin. Here you can see that there is still a line of nucleus, but there are more concentrated parts of the chromatin, and they are called karyohectic cells, and they represent the late apoptosis. Here we are coming to necrosis. So this is a pycnotic cell, really shrunk nucleus when you compare it to the cytoplasm and to other nucleus. This is the ghost cell, so it doesn't have the nucleus anymore, and it is called the karyolytic cell and represents the late necrosis. This is actually the differentiated mononucleated cell with micronucleus. So they are in the same focus, the size is okay. This and this, This is actually the broken egg. So those are two parts of the nucleus connected with the bridge. But if you connect them together, they will make the oval shape of one nucleus. That's why they are called the broken egg. And this is actually the nuclear bud. So we are repeating once more that apoptosis is the programmed cell death. When cells are too damaged, they kill themselves. And necrosis is normal cell death, usually when cells are old, in senescence, etc., etc. There are some structures here that you cannot, that you can see, but you cannot measure them because, for example, this is binucleated cell, but it has the nuclear bud. So this type of DNA damage will not be checked in your list. So you can just say that this is binucleated cell, but you will not talk about the nuclear bud. So this assay still needs validation, standardization, and automation. And this Human XL group in this year has published 2 review articles about their achievement in these 26 years from the beginning of the use of the buccal micronucleus assay. And you can check both articles if you're interested to see what are their what are the improvements of the techniques. And they have also published the other review article about the use and implementation of the buccal micronucleus cytomassay in human studies, but I will tell you more later about that. For validation and standardization, scientists and labs, they need to be trained. So this group has performed 3 interlaboratory scoring exercises, and between also experienced and non-experienced lab. They had 3 referent labs. One was in Australia, the other in Italy, and the third in Austria. And they have concluded that even unexperienced scientists, that means unexperienced laboratories, they can have a good number or the frequency of micronucleus that they have counted, but that they still have problems with, for example, apoptotic and necrotic cells, especially for apoptotic ones. So they have concluded that those labs still need to exercise more, or that the apoptotic, late and early apoptotic phases should be connected together. Now let's talk a little bit about the Comet assay in buccal cells. So this technique is not so old like the buccal micronucleus assay. It has been developed from the actually in vivo mammalian alkaline Comet assay that has the OECD guideline 489, but in vitro is still not validated. Comet assay can be done in 2 ways, depending on the type of the damage that you want to check. And double-strand DNA breaks are measured in neutral Comet assay, and single-strand breaks and alkali labile sites are measured in alkaline version. For the Comet assay, you need the single-cell suspension. And those cells, after the single-cell suspension, they need to be in some kind of sandwich of agarose gel layers on the microscopic slides. Usually, you are using 3 or 2 layers. The lower one is without the cells, and the second one is the mix of the agarose and the cells. And after solidification, these microscopic slides with the cells go into the lysis treatment that removes the cell membranes, cytoplasm, and nucleoplasm, including nucleosomes. You can also make different specific types of the alkaline Comet assay, depending on the DNA damage. For example, oxidative DNA damage with FPG or other types. And then after the lysis solution or lysis treatment, you are using the treatment with those enzymes. After that step, there goes the denaturation, either neutral with pH 10 or alkaline one with pH 13. And this denaturation serves for unwinding of the supercoiled DNA so that the nucleus is relaxed. So if there is some kind of DNA damage when cells or microscopic slides go to electrophoresis after the denaturation, these fragments can go through the electrophoresis field, while the nucleus cannot go because of the size of the agarose pores. And then after staining, you can check or assess the DNA damage either visually with making different categories of the damaged cells that have the shape of the comet if you are using the alkaline version, or the shape of the foggy, or you can use the softwares. We have done many experiments and many protocols, and in our lab, only the neutral comet assay from this protocol had success. So we are usually measuring this kind of damage, but we have also published this manuscript where we have described how we have compared different protocols. And on this picture, you can see what are the types of the damage or the tails when you are using different time for the lysis, so proteinase K, trypsin, etc., that are usually used for the buccal version. Because here the biggest problem is how to remove the cell membrane, that means cytoplasm, and to relax the DNA, so to get only the nucleus. The solution maybe for this problem is to use the comet assay in the buccal cells. As I have already told you, all the cells in your mouth are called the buccal cells. So we were talking about the exfoliated ones, but in this protocol, in this manuscript that you can see here, they are using, after the saline for the mouthwash, saliva. And in this saliva, you can see or get the buccal mononuclear leukocytes, and in them you have mostly lymphocytes. And alkaline version of the comet assay really works every time in this protocol. So the pixels are not so good, but you can see what are the problems in the buccal comet assay. So after the electrophoresis, there is no DNA damage if you don't have the DNA that is free and relaxed. So here is the cytoplasm that is still stopping us to see the damage of the DNA. Here we have managed to destroy a little bit this cytoplasm, but still the DNA is trapped inside. Here are still parts of the cytoplasm, although you can see the cloudy nucleus. So these pictures do not represent good cells for counting, and you cannot measure the DNA damage in them. In those cells here, you can see that this cell is relaxed and that there is a cloud. Here, there is no cloud, and that means that although you cannot see the cytoplasm, there are still some things that are attached to DNA, so that after electrophoresis, this nucleus are still not relaxed. And this cell is also okay, so that means that we were able to remove the cytoplasm. So that is the problem with the buccal comet assay. So this assay also needs validation, standardization, and automation. And there is another group that has been made that is called COMNET group. And this COMNET group has even started one cost action that was named the COMET assay as a human biomonitoring tool that lasted up till 2021. And in this assay, the scientists from more than 70 labs all over the world have gathered together. So they have made a lot of protocols, a lot of validation. They have published more than 120 articles and have made a lot of training schools. And after the finishing of this COMET assay, where they have made validations, Now they are gathered in a group called International Comet Assay Working Group under the supervision, let's say, of the European Environmental Mutagenesis and Genomics Society. And on the web page of this EEMGS Society, you can check the protocols, the news, and everything about this International Comet Assay Working Group. Also, International Comet Assay Working Group has their YouTube channel, where you, if you want to implement Comet Assay or some types, specific types of the Comet Assay, or if you have some troubleshooting, there are videos, short ones, that step by step explain to you how to perform Comet Assay, how to prepare the slides, how to make assessment of DNA damage, even visually, or with the softwares. And you can check this YouTube channel. There are evidences that are connecting also the Comet assay in lymphocytes or leukocytes with the prediction of risk of death and cancer and mortality. But since the buccal Comet assay is a more newer method, it is still early to make a connection for the buccal Comet assay with that. But with the new manuscripts and the results, in the future, we will be able to make new meta-analysis and make new conclusion about the use of the buccal Comet assay. But right now, this buccal Comet assay cannot be used to disease risk assessment, but it can be used. There are enough evidence for the risk assessment after the exposure. And most of the articles in the literature are connecting the DNA damage with the exposure to the metals in the dentistry, in the farmers, but farmers are usually connected with the exposure to the pesticides, with smoking and vaping, with air pollution, X-rays, with the diet, with infections, and some specific types of work like jewelry workers, painters, and the people that are working with anti-neoplastic drugs. In 2019, we have made the first literature review, and now in 2024, we have done another literature review. So I'm showing you some results from the Web of Science, how the interest of the scientific world has increased in those two essays or in the buccal cells. So from the 2019, from 3,000 to 6,000 articles have been published about the buccal diseases, 10,000 and almost 700 about oral epithelial diseases. So there is a big increase in scientific interest in that, because you also need to know that for the cancer risk prediction, it is important. Epithelial cells are important because more than 90% of cancers are of epithelial origin. So if you detect the DNA damage or elevated levels of DNA damage in epithelial origin, you can inspect those people or even predict if they will or not develop the cancer, which type of the cancer, and you can make a therapy to prevent those things in time. Buccal DNA damage, almost 800 articles nowadays. In buccal biomonitoring, almost 200. In oral epithelial Comet assay, 78 today. And in buccal Comet assay, a little bit more than 200. Because the Comet assay is not so old technique. But for oral epithelial micronucleus, you have almost 300 articles now. And if you can see, 900, almost 1,000 articles that are using the buccal micronucleus. So as I have already told you, the buccal micronucleus can predict the risk of carcinoma almost linearly in oral diseases, so the same origin as the buccal cells, and it has been demonstrated for oral squamous cell carcinoma, gingival or tongue cancer, and squamous carcinoma of larynx, pharynx, and oral cavity, and most of the head and neck cancer frequencies. In those cases, not even the micronucleus frequency was increased, but there was also a higher frequency of binucleated cells and cells with apoptosis. You can check this article for this systematic review because those are results from that systematic review. Buccal micronucleus assay was also able to predict DNA damage and the risk in oral pre-malignant mouth diseases and also non-malignant mouth diseases. So something like leukoplakia or lichen planus, but also, as you can see, periodontitis and stomatitis. They have even been able to predict risk of bladder cancer and breast cancer patients. And in 2021, there was another new review made that has explained how buccal micronucleus can be used to check the risk of upper body cancers, as we said, head, neck, and breast cancers. And you can check also this article if you want to learn more details. In 2024, based on all those evidences for the scientific literature, it has been concluded that buccal micronucleus assay can be used also as a clinical biomarker of genetic damage in all types of oral carcinogenesis. So you can also check this article if you're interested in more details about that. So we have talked about the cancer and oral diseases, but buccal micronucleus frequency was able also to detect or predict the development of diabetes, some diseases that are not connected with oral cancer, and that is diabetes, arthritis, even lupus erythematosus, and Crohn's diseases. Also, in Down syndrome patients, there were really elevated levels of buccal micronucleus frequency, especially in the younger Down syndrome patients of 8-fold. Also, in Alzheimer patients, there were elevated levels of not only micronucleus, but also there were changes in frequency of the basal cells, condensed chromatin, and karyohectic cells. And in buccal cells, there was even possibility to measure the tau and tau levels. And you know that usually tau is measured post-mortem from the brains of the people to actually confirm that they had Alzheimer's disease. So you can measure the tau levels in live patients, and that is a really important thing. As I have already mentioned, the diabetes, there were also increased frequency of buccal micronucleus in anorexia nervosa and bulimia nervosa patients. In our one of our manuscripts has also explained the connection between exposure and development of the diseases in asbestos, current and former exposed workers. We actually had 600 individuals, but because some analyses were not successful, at the end, our manuscript called for 186 people, and we have demonstrated elevated levels of the formerly exposed, the micronucleus frequency in the formerly exposed people. And what was really interesting, that we have demonstrated that 12 years of exposure, duration of exposure, is the most important thing in the risk assessment, whether you will get asbestosis in one moment of your life, because you never know when asbestosis will come or will it appear at all from after the exposure to asbestos. But if you were exposed for 12 years, no matter when, or if you're currently exposed and were formerly exposed, for sure you will get the asbestosis. Here is again the list of all the lifestyle factors that were connected with the elevated levels of buccal micronucleus frequency, but we have already mentioned most of them. And of course, smoking and alcohol intake have a big influence on the increased buccal micronucleus frequency, together with some exposures to pesticides, anesthetic gases, arsenic, and some kind of diseases that we have already demonstrated. And this is the article that has collected all those human population studies and analyzed them statistically and epidemiologically. Micronucleus frequency in the buccal cells has been connected and correlated also when in the same individuals were used other types or assays of DNA damage, such as lymphocyte micronucleocytome assay, chromatid aberration or chromosome aberration, and sister chromatid exchange assays, and they have all demonstrated the connection or increase of DNA damage in all those assays in mostly people that had smoking, different smoking or tobacco chewing habits, as you can see here. With radon, with exposure to chromic acid, ethylene oxide, benzene, or leather tanning chemicals. Lead, arsenic in drinking water, and exposure to the pesticides. So the line of the achievement from 1982, when the buccal micronucleus assay was first published and started to be developed, you can see in this timeline. So today, or in this, as I have told you, there was a detailed protocol, comprehensive set of detailed scoring criteria. There were inter-laboratory site scoring exercises, and there was a series of systematic reviews and meta-analysis on the connection of buccal micronucleus frequency and diseases, because this assay and the scientists that are interested in this assay, they want to make it important and established biomarker in disease prediction, cancer prediction, and as a clinical biomarker. But for all those things, there are still, there is a need for more articles with a bigger group of patients, with the use of more DNA damage assessment assays used together in the same individuals, so that we can perform new meta-analysis. And there is still place to validate more, standardize more this protocol, and make it automated so that it is much more faster. On YouTube channel from the ECOG, you can also check some information about the micronucleus assay. And micronucleus today as a clinical biomarker is currently used in radiation biodosimetry, in assessment of the exposure from the chemical genotoxin, in deficiency of folate in vitamin B12 and spleen function. There are enough evidence to translate it into clinical practice for prospective cancer risk and staging of cancer, cardiovascular mortality risk, accelerated aging, and prospective pregnancy complication. And it's still inadequate to be used in prediction of neurodegenerative diseases, fertility, viral infection, and frailty. In 2024, this Human XL group has demonstrated that all those DNA damage in all those types of the organ can be connected, and that oral mucosa, and that means buccal micronucleus, can be used to assessment of the exposure and the risk assessment of diseases after the airborne and dietary genotoxin, and that is also connected with glycation and inflammation. And if you don't have the lymphocytes micronucleus, you can use also cells from other types of the organs, such as kidney, urine, and bladder, and cervix, to assess the DNA damage. And this can be translated also on other organs or in organism in general. So in conclusion and perspectives of those two assays, so there is still a lack of prospective studies that can show the elevated levels of micronucleus frequency in buccal cells and how this can predict an increased risk of developmental degenerative diseases. There is also a lack of knowledge what the basal cells and the mitotic rate in basal cells can do to the levels of DNA damage detected as the micronucleus frequency. There is a lack of automated systems to score the micronucleus frequency in buccal cells, although there are some softwares in use that are using the DAPI staining, but with DAPI staining then you cannot see the cytoplasm. And you can also check only the micronucleus frequency, not the other types of the DNA damage. And the damage that is detected with those automated systems is lower when you are assessing the micronucleus frequency with by yourself with the fluorescent microscopes. There is a lack of image analysis algorithms so that you can check other different types of DNA damage. Not all the, considering the buccal comet assay, not all the laboratories are successful with that. So although we said that considering the micronucleus assay, the buccal one, there is still a need because the micronucleus is used in general to differ the healthy group and disease group, but still there are some differentiated disease variables that are important so that you can also use it on individual level. And we should use the prospective cohort studies and randomized controlled trials to verify for which diseases micronucleus can really be precise in prediction and how micronucleus frequency can modify or alter the disease outcomes. And we should always use the CITEM approach because CITEM approach was not so often used in the past, but nowadays almost all the manuscripts are now using the CITEM approach. I would also make my special thanks to my colleagues, that is Goran Gajski and Marko Geric from my Division of Toxicology at the Institute for Medical Research and Occupational Health, Zagreb, Croatia, that are part of our ToxLearn4U group. Stefano Bonassi, that is also a part of our group, and he's from Clinical and Molecular Epidemiology in San Raffaele, Rome, and Claudia Bolognesi from Environmental Carcinogenesis Unit from the Hospital Policlinico San Martino in Genoa, Italy, and her lab was one of the referent labs where I also went to improve my knowledge, my scoring techniques, and she has published many articles with a lot of photo galleries to help new scientists and unexperienced scientists to become better in the assessment of DNA damage with the buccal micronucleus assay. And I would like to thank you for your attention. If you have any other questions, you can write me on my official mail that you can see. And we will make two small exercise for you where we will give you more details on the manuscripts that you need to read online and then answer on some questions after the reading those manuscripts. So thank you once more.